from the genomic mapping, we can easily understand any kind of a gene where it is present in the genome. Now, okay, let's understand what is the genome. Genome means is a collective of a gene that is distributed different type of a chromosome. In case of a human, female, they have a 23 different type of a chromosome, and the male, they have a 24 different type of a chromosome. And in general, human have a approximately 29,000 genes. So this vast number of a gene, it is distributed this different type of a chromosome. Now from the genome, we can easily say this gene is present in this chromosome and in this location as a pinpoint. Now how we can determine? For the determination of the chromosomes location where gene is present, we used to take different type of a, we used to take help different type of a techniques. One types, okay, so before that we can differentiate two way. One is the genetic mapping and another one is the physical mapping. Under genetic mapping, the technique most frequently used, it is the recombination techniques. Any two different genes, they recombined very frequently. That means these two genes are present distantly. That means they are located, or the distance between them is more compared to the those two genes are recombined very less frequently. We call them as a linked or the linked gene, because their distribution or the separation is very less. So on that basis, we can say the arrangement or the distance between the genes. But here you have to understand one thing. We are talking about the distance in the sense how far they are seen. So we are not talking about any kind of a scale, any kind of a physical process. We can identify, or we can say this much distance they are present. Instead of that, we here denote as a centimorgan, right? If there is a 1% frequency, that means 1 centimorgan. 10% frequency, that means 10 centimorgan, like that. So this is one type of a method, and another one is the physical method. Here we will talk about the actual distance. Like, to understand any kind of a, if 2 points in a paper, to understand how much distance is present on that paper, the 2 point A and B, we used to measure with the scale, like 1 centimeter, 1 millimeter, 10 centimeters. So we used to take the help of the scale, right? So there is a one unit. Same things, that means we used to use any kind of a physical object to understand how much far between A and B. So in case of a gene, we can't use any kind of a scale. Instead of that, what we used to do, we try to count how many nucleotides are apart between any two genes. So that's why here the unit is a nucleotides, and that's why the name came as a physical mapping. So for the determination or of the distance of the two gene, we can use different type of a technique, like RFLP, restriction fragment length polymorphism. Means we used to partially digest the DNA and or the chromosome, and after that we used to run the gel, and on that basis we understand or we determine how much distance is present. So with this, we can specifically say between these two genes, there is a 1000 nucleotide apart. So between these two genes, 1000 nucleotide is present, or the 20,000 nucleotides are present. So that means we are quantify how much distance they are. So that's why it's called them as a physical distance. And in physical distance they actually say the real distance, but in a genomic map we can't specifically say how long or the how far these two gene is present. So this is the very basic difference and very important and the easy difference. So one is the physical mapping and another one is the genetic mapping, and in this way we can determine the genomic map or to establish or to construct the genomic map. For more video, please like and subscribe.